Yeah, welcome.
We should be, I'll another few seconds and we should be live on YouTube also.
Perfect.
I'll
Ma'am,
let you.
okay. Ma'am, I'm not a host as of
I'll
now.
make you.
Yeah.
I'm co-host. Yeah, ma'am. Sorry.
Live on. We are on YouTube.
Ma'am, okay. Ma'am, I'm not a host as of now.
Making.
Yeah. Sorry.
We are on. Making.
Shall we start? Very good afternoon, everyone. We warmly welcome you all to this Journal Club of our Academy of Clinical Embryologists of India, where we continuously strive to bring international standards and evidence-based discussion into our IVF laboratories. Through these sessions, we aim to address relevant day-to-day questions in embryology by learning directly from eminent scientists and global experts. And as a part of this series, today we'll focus on a very practical and often debated topic, that shelf life of a culture media. In our IVF laboratories, we are always concerned about the culture media quality, stability, safety, as these remain the matter of prime importance for all of us. But with the advent of single-step culture media with longer shelf life, a common question bothers all of us. That is, how long can these media be used safely and effectively? While product label permits prolonged use, we often wonder, is it truly safe to use media close to expiry, and does longer shelf life impact performance or the outcomes? And these are real concern in everyday laboratory practices, and addressing these query requires strong scientific evidences. And we are therefore delighted to have Dr. Dean E. Morbeck with us today, who has recently published an important study in Human Reproduction in December 2025 issue addressing this very issue. And it is privileged to have him to present his paper himself and guide us through the data and its practical implication for our laboratories. To discuss this very relevant paper with Dr. Morbeck, we have Dr. Sumna Gurnath with us. We have her slide. So we have Dr. Sumna Gurnath with us today. She is consultant in infertility and reproductive medicine at Cloud9 Hospital, Bangalore, with over 23 scientific publications and 8 book chapters. She also serves as an associate editor of the Journal of Human Reproductive Sciences. She brings a thoughtful, evidence-based perspective to embryology, and I'm sure her insight will add great value to this discussion. Along with her, we are joined by Dr. Nishad Chimote, a leading embryologist, known name in our fraternity. He is director of Vanshdhara IVF Center, Nagpur, a managing committee member of ACE and incoming vice chairperson of ISAR, widely recognized for the practical technology-driven embryology training. We are really glad to have him as a part of today's panel. Along with him, we have our ACE Vice President, Dr. Gaurav Majumdar and Dr. Parsuram Gopinath to chair this session, the two stalwarts in our field. Dr. Gaurav Majumdar, he is currently working as a scientific director of Sir Gangaram Hospital, New Delhi, chief clinical embryologist as well. And he is having more than 20 years of ART experience, vice president of ACE and past chairman of embryology at Indian Fertility Society. His core expertise include KPI, data management and quality systems in IVF. The another chairperson is again a very known name in our fraternity, Dr. Parsuram Gopinath. Dr. Gopinath is our dynamic secretary, medical and scientific director at Simar chain of hospital across Kerala, Coimbatore, and nationally decorated clinician with multiple awards, over 10,000 ICSI cycles and landmark achievement of establishing Maldives' first IVF unit. With such an expertise and diverse panel, I am sure today's discussion will be insightful, practical, and very relevant to our laboratory practices. So over to you, our chairpersons. Please welcome and introduce Dr. Dean Morbeck and start the session. Over to you, Dr. Gaurav.
Right, right. So Parshu, I'll go. And so I'm really glad to have Dr. Dean Morbeck join us for the ACE Journal Club. And he's a very well-known scientific figure in India, especially here. And we've listened to his lectures in our conferences, and we always look forward to the knowledge that he disseminates. He's the Chief Scientific Officer at Genia Fertility um, in Australia, and he's also the Adjunct Associate Professor at Monash University, Department of Obs and Gynae. He's also an honorary lecturer at the University of Auckland in New Zealand, and he's part of many advisory boards like Apricity, ART Compass, the World Egg Bank, and Vivoplex. In addition to this, he also holds a MBA as well as a PhD in physiology. He has various publications, like I said, and he's been one of the major contributors to the Istanbul Consensus. So over to you, Dean. We are eagerly waiting to hear.
Right. Thank you, Gaurav, and thank you all for the invitation and for taking time to join in. I look forward to discussing this paper. And Manisha, I agree. It's one of the questions we've always wondered about is, can we use media to the end of its shelf life? And I'll share my journey that made this be something that I could study. First, I do need to clarify on my disclosures there. That wasn't exactly right. There was prior ones. They were correct. I'm still at Genea and at Monash, not at University of Auckland. And my main conflict here would be that I work for Genea Fertility, but Genea Fertility is no longer associated officially with Genea Biomedics. They're two completely separate companies. So we have a strategic partnership, but we don't have any link. There are actually, Genea Biomedics is owned by a company in China called BaseCare. So it's no longer the same. So even though you see the word Genea on both, we're not connected anymore. So it's a disclosure that I do still, if they want me to give a talk, I'll give a talk, but we don't do more than that. So with that, I will start, let's see, find my... There we go. Okay, is that visible? And can you see my pointer?
Yeah, it's visible.
Okay, good. So this is a journal club on this paper that is in press currently last month and here at the ACE meeting, the ACE Journal Club. So to start with, it's important to give you kind of my culture media journey because my views on culture media have evolved over time. So when I first started IVF back in 1995, I was buying powdered HTF from Irvine and reconstituting it and making our own media, filtering it, doing all the testing on it. And that was here in Ohio, this red state here. I actually was doing my training in Minnesota, which is this state here, and I did my graduate work here at North Carolina. That's why that one's highlighted. But so Minnesota and Ohio are the two key players during this time because I was in private practice. And it was a key time because it was also when I really saw that the quality control of the products we use is really critical to our success. And I realized that it needed to be better. And so that 10 years of in private practice then translated me to going to Mayo Clinic in 2005 or so, and then really studying a lot about quality control, culture media, protein, many different topics. Here's one of the papers where we re-engineered culture media. And then, so as far as my culture media journey, started with HDF. Then once it became like not practical to just make it yourself anymore, we switched to something else, and we switched to Sage Sequential at the time. But I was only doing cleavage stage culture and up until 2004. It's a whole nother story about how I converted over doing blastocyst culture, and it's pretty fascinating in and of itself. But nonetheless, I got to Mayo Clinic. They were doing cleavage stage transfers, 2PN freezing, actually, zygote freezing, and we were using G1, G2, and they've been using that for quite a while. Actually, it couldn't be all that long because it came out in like 2000, so, or 99, somewhere in there. So they had bought into it and were using it. And then once we got going into blastocyst culture, we converted to using global total. Now in the US, we reconstitute with our own protein supplements, so it's very different. But this is the time where I said, okay, I now have the resources and the students and everything to be able to do more research into quality control. I then took that once I finished my MBA and went to New Zealand over here in this tiny little spot over here in 2016. And I inherited what Tex Vermillier had put in place right before me, which was he switched from G1 to CSC. And so I came in and I already knew based on this paper that CSC and global were very similar. So I was like, okay, I'm happy with CSC. We'll continue with that. We did try to switch to GTL, and that was chronicled in this paper. It didn't work for us. I still think it could be a very lab specific issue there, but we switched back to CSC, and then we did another study where we looked at CSC NX, and we saw an improvement again, and so we stayed with CSC NX, and that's what they're currently using in New Zealand. In between here, I went to New York and worked with another company, but was pretty far removed from the actual day-to-day. And then in 24, so now 2 full years, I've been at Genea in Sydney, and we're using Jerry single-step media. So you can see I've used a lot of the different culture media that are out there, many that are still being used. And I'm currently using this, and I don't have any plans to change because it works well. I'll show you the data on it. But I don't know that we're at the end of the story on that. And I'm always interested in doing comparisons and seeing if something new could improve. Like the GTL Ox was something I thought was intriguing. The results haven't been all that impressive, so it's not one that I'm currently looking at. But you know, those types of developments I expect will come along. Now, I need to give you a little bit of the history of this medium because it matters what it is. So, what most of you probably don't know is back in the 1990s, Genea was called Sydney IVF, and they rebranded right about here. But in the 90s, when they were Sydney IVF, David Mortimer was the scientific director. He developed sequential media. It was sold by Cook. That's the Sydney IVF culture media. And then eventually Sydney IVF said, you know, we're having Cook do all this. Let's start doing our own. Let's start doing our own R&D. And so then they made some changes. I don't know what the changes are because these culture media weren't available to me when I did my reverse engineering. So I couldn't study these. And I still don't know the composition because I don't work for Genea Biomedics. But for these years here, Genea Biomedics or Genea Fertility was using sequential. And then in 2016, the 10 or so clinics switched over to Jerry single step because 2016 was the year that they started using time-lapse, the Jerry time-lapse incubators across the board for all cycles. And so here's that little bit of, you know, change that occurred. Genea Biomedics, which was a company within the Genea Group, then was sold out to a company called BaseCare. So now they're completely separate. So what I didn't know when I got to Australia, when I got to Sydney 2 years ago, I did not know that the shelf life of this media was 1 year. I knew that shelf life varied across different culture media, but I didn't really pay much attention to it because I chose the culture media I liked and I just went with it. But it was probably about 8 or 9 months into my time where all of a sudden I'm like, wait, we've got a shelf life of that long and we've got data like really good like lot tracking data that we can say we know which lots were used so we can really look at see was the age of the media important? I said, well, maybe it is. Maybe, my belief system has been that, yeah, media, you don't want to use it towards the end of its shelf life because it might go off. We don't want to take chances. So here I'm sitting with the opportunity to look and actually not only for one, test to see the theory that maybe it doesn't matter, but the driving force for me was I was like, what if it does matter? What if we're affecting care and we need to change that? Like I don't know what biomedics would do with the information, but I know what I would do with the information. I would say we're cutting it down to say 6 months if that's what we found. So that was really important to me to study this, and thus it resulted in this publication where we were able to look. And these are a couple of the embryologists at Genea, long-standing, been there for a long, long time. And so, they were instrumental in making the systems in place and the lot tracking and all. So we did this, and now it's been published. So you hopefully all have access to it. And I'll just cover some of the background on it. So one of the things that you would know if you're using different culture media is that the shelf life isn't the same. And I'm just looking at single step. I'm not looking at any of the others here. But you can see the different manufacturers. This is by no means all of the different culture media manufacturers, but these are the most common ones that I've been exposed to and used. So Genea's got 12 months. Cooper Surgical, the global total is 10 weeks. So that's what, 2.5 months, but Sage One Step is 6 months. So interesting, obviously, because they came from two different companies when Cooper bought them. GTL from Vitrolife has different ones depending on the country or the regulatory body. And then Irvine, which is, I had it here as Fujifilm. It's no longer that. I think it's Next Spring now that would be there, but that's historically where I got that information, and that's sitting at 4 months. So, we've got as short as 2.5 to 4 to 6 to 12. So quite a wide range there. So that presented this idea that, okay, what's the research context that we're sitting here? We have established parameters for culture media. While the manufacturers don't tell us the exact composition, between myself and the 2 studies I've done, and then 2, maybe 3 studies now from Sebastian Masterbrook's group, we know pretty well what's in the media. It'd be great if the manufacturers just told us, because we all know. I mean, just tell us, but that's another story. So we also know what our pH is because we test that. We know what the osmolality is. These are all studied and well controlled. What we don't know is the stability of the culture media. How good is it? How long does it stay good in the on the bench? And we also know that there's this variation of roughly 2 to 12 months across the manufacturers. So the research question for us was based on, I came in and saw, wow, we've got all the data and no preference. It wasn't like there's ever been a change where they said the shelf life changed, so we couldn't do before and after. There was never a time where they said, oh, we're going to try to always keep it at 6 months, but sometimes it might go longer. It was pretty random across the entire time period that I'll show you. And so it was just natural use of a product that had a 12-month shelf life. And we had good records to be able to track what the outcomes were. Now, a little bit of background about what we think might be going on with shelf life based on just a couple of papers. And these are the only 2 papers on the human culture media. So again, the Dutch group looked at the age of G1 plus, V5. Now this is just cleavage stage. And it goes out to the, roughly 3 to 4 months. And they saw a decline in birth weight with the age, a sample size of 372. So an important point. That's a very small sample size to look at birth weight effects, given all the other variables that come into affecting birth weights. So I'd say that of course gives us a concern. We say, wow, well, that should be followed up. That was in 2015. It's now, she's 11 years later, and it hadn't been. Massenburg's group did another study, Terahome, where they looked at what happens to culture media, does it break down? And there is some change. Some of them, these are all the different metabolites on the x-axis here. And the y-axis is 100%, and you can see a lot of these amino acids actually increase somehow in concentration, fivefold. Not sure how. But we also know that glutamine is one of the more labile things, and that will decrease as it will break down a little bit. Although all of these culture media now, or at least most of these, I think over here, use the dipeptide glutamine. So if you're not familiar with that, glutamine, because it breaks down and creates an ammonia byproduct, the manufacturers link it to another amino acid, and they all use different ones. But a common one is alanine, glycylglutamine. Those are the different ways. And so if that does break down, you can imagine that the other amino acid, alanine or glycine, would go up. And here's alanine that goes up. Okay, so if it's breaking apart, then that's would explain this one. So while this is, I don't think we can dispute that this probably happens. What we don't know is it clinically relevant? Does this matter? So here's the study design. It's a retrospective. It had multiple centers. It was over a period of 4 years, around 3 years, about 10,000 cycles, but only 6,000 patients, or not only, but 6,000. So you can see there was repeated measures there, 8 sites. The culture system we used was the single-step medium throughout. It's the only medium we used, Jerry. We use time-lapse incubators for all cycles, and there's no media refreshment. So they go all the way to the end of the culture period. Now, the age range of culture media, we had some that was as young as 38 and some that went right up to its expiry date, 365. But the mean was about 190 days. And what we saw was it distributed relatively normal across that, where you'd see a peak in the middle and then coming back down. I forgot to put that slide in to just show that it's, the most common was right around 6 months of age. So already further than most of the other manufacturers. And then we looked at outcome measures like embryo development, so blast use rates. We looked at the cumulative pregnancy rate. So not only the first pregnancy, but what's the cumulative pregnancy and cumulative live birth rate. And then importantly, because of that paper on G1, that we wanted to see if there was any impact on birth weights. Now, because of the nature of the data, we needed to obviously do regression models of all the different variables, and then also GEE accounting for that repeated measures. All right. So here's all the results that I'm going to show, because it would be boring to go through every single one of these different slides. In essence, we saw no effect. Now, you're right away going to look and say, no, there's got to be an effect. That first one's really low. So you can say, wow, if you get the media too early, it might not be ready. Maybe it needs a little, like wine, it needs a little, aging. It's only 105 cycles, so that's clearly a sample size effect. Every other one of these is in the thousands. And so you can see, though, the distribution. It's mostly between 3 and 9 months, but there were 1,100 cycles that were 9 to 12 months. And then you look at all the different variables, so the day 5, 6 blasts, the usable blasts, all the pregnancy outcomes. Importantly, the birth weight was not affected. And then we did subgroup analyses, PGT cycles, fresh transfers, all transfers, looked at it every way we could to see, are we sure that there's no effect? And we didn't find any. So that was very reassuring, because I really did think, when I started this, I truly thought we're going to be cutting back on what we use. We're going to change our use pattern, because I believe that it would make a difference. And I'm glad to have been proven wrong on that. So this is again the same, I guess, just a different one. No association with media age. I, for some reason, have a duplicate of that slide in a different format. So just maybe a little bit less messy on that one, but same results. So if we put that into the context, so we've got the Kleiker's paper from 2015, which gave us an inkling, a concern that birth rate could be affected. Animal studies give us conflicting results on this, on the effect of storage. So really, we have so little human data that we really don't have previous literature. So then there's our findings in that face of that. And what our findings are for one that, this is a single step media up to a year. There was no birth weight effect. There were no pregnancy outcome effects. And this was basically all blastocyst stage transfers. So there was, quite an exposure time. This wasn't like cleavage versus blast. What we believe is going on here is that there's, for one, again, I alluded to it before, the biochemical stability, and that's these stable dipeptides. So having L-alanine, L-glutamine instead of glutamine alone is one of the key ones that really, that just came out, well, I'd say just, time flies. That's been in existence for more than 20 years now. I mean, obviously it's been in existence for longer than that, but it's been in culture media for more than 20 years. But when you go back to the old days in the 90s and the 80s when they were studying, really trying to solve the different developmental blocks and all the different issues, glutamine was one of the key components that they had to be careful about because it was so labile. So the clinical implication of this study is that the extended shelf life single-step media maintains complete undisturbed culture performance throughout the 12-month storage. So that's good news for, obviously it's good news for biomedics because that's what they banked it on, but it's good news that I didn't have to change anything and we get to talk about this. So just putting this in the context of, the Escher Good Practice and IVF Labs 2016, this still depends on a lot of good practice. This isn't just, the meat is fine. No, we still have to have good storage conditions, that we maintain that 2 to 8 degrees, that we have really good SOPs that we follow. The post-opening handling protocols. Now this, I know, varies a lot, and I'll talk a little bit more about it. But, and also how long you keep it once you open up a bottle, which is pretty standard to be, some people keep it 5 days. Most, I think it's a week. But then that we do all of these regular QC monitoring media tracking system. That's really key components of laboratory excellence. Again, the findings listed here. And just a very important note, this is for one culture media. And so we can't say for sure that this applies to other culture medias. And obviously it would take the manufacturers to take the steps to prove that and then change the shelf life that they have listed for that. And there are significant differences among the medium, but I'm not going to talk about the different, components in the culture media tonight. What I do want to end on, but include, is that Janae has been using these culture media now, either as Sydney IVF or as Janaea, for a long time. In 1997, it was a banner year. That was when they started using their own sequential media that then Cook started to sell. And that year, their pregnancy rates went up considerably. And so clearly that was a benefit. They also started using Mink, the mini incubators, the Cook Minks, which they weren't Cook yet, but Cook then started selling those as well. So they used that throughout this period. Now, it turns out that I've got electronic medical record database that goes all the way back to 2012. So I have all of the transfers, the retrievals and transfers for this period. And so I can look at what happened for these years. So there was like a 4-year period, 2012 to 2015. And then they switched to time-lapse and Jerry Medium in 2016. So I can't say that this isn't about, is Jerry Medium better? This is, have the changes that were made at Janaea, did those have an impact? Did they change? We can't say which changes when it mattered, but they made a lot of changes in 2016 by going to time-lapse. Now, when I say time-lapse, another thing that is pretty common in the world is they only used it for time-lapse. They only used it for incubation. the time-lapse incubators. They did not use it for embryo selection. So the important note, right, that they didn't see a benefit if there is one with time-lapse selection. Again, I think I talked at the ACE meeting, and I don't remember where it was. It was at least 10 years ago, about that I don't think it's as important for embryo selection. It's more about culture quality. So they made that change. And so I could look at this and say, well, was there any impact? And you say, well, what variable would you look at? Well, you could look at blast rates, which I did. But then I thought, the most common way to look at patients' prognosis is to look at their live birth rate over 3 cycles. And we're lucky enough that we live in a world here in Australia where patients are getting funding that they can do 3 cycles routinely. Though many patients drop out, 30% of patients drop out before they're successful or do 3 cycles. But this is a common thing, and I'll show you here. This has been published on a lot. So here you can see like 5 landmark papers, New England Journal of Medicine, JAMA, HR, and Medical Journal of Australia. But this one, ITT, means intention to treat. So this is taking, let's say there's 1,000 patients that start, and how many got pregnant? And it has no age, so these could all be different age groups, but this is across a big population, no difference in age. And you say, well, if it's 45%, that means 450 out of 1,000 got pregnant after 3 cycles. Now they might have dropped out after 1 cycle and never tried again, and they'll count as a negative. So that's intention to treat. This optimal level is something that is a statistical measure where you say, well, what if that patient that dropped out after 1 cycle stayed in for 3 full cycles? Some of those would have gotten pregnant. And there's a statistical measure called a Kaplan-Meier estimate, which takes the average rate for each cycle and then applies that to the M, and it gives you an optimal chance. So you can see that the optimal chance is higher. The reason why I have this here is because none of these studies used cycles after 2012. So they were all back in the days when there was a lot of cleavage stage transfers and a lot of double embryo transfers. Now I know that topic is very regionally, geographically, and socioeconomically varied across the globe. But we know that there's a lot of places, Australia being one, that does single embryo transfer predominantly. So we have a twin rate that's much lower than this 20 to 30%. And twins carry a lot of comorbidities, so we really think we should avoid those. So this doesn't really represent the current state of at least how we're practicing in Australia, which is why I then went ahead and said, well, let's look at how does this culture media that's got this year shelf life do compared to these, and also compared to the sequential media that came before. So here's the where we implemented that Jerry continuous media in 2016. So that's this is data across all 10 years. It's 18,000 patients. And you see it for the first 4 years, or first 5 years. Blastocyst utilization was 48%. The optimal cumulative live birth was 67%, and they both went up. It really jumped dramatically for blast utilization when we switched over to time-lapse and Jerry medium, and we went up to 70% for optimal cumulative live birth. So it's an increase in cumulative live birth after that switch, and a 19% increase in blast utilization rate. These results have been submitted multiple times now, trying to get a journal to take it on. So when I update that evidence from that previous graph, you can see that here, it's really pretty remarkable. We've got better outcomes than the historical ones, which were transferring more embryos. We're using, so this is on a per patient coming in basis, 57% go home with a live birth. And 68% is the estimated amount if you didn't have dropouts. And this is with 97% single embryo transfer, 3% twin rate. That is a huge decrease in what is really not a safe outcome to have twins. So I think that's a pretty remarkable confirmation that the shelf life story isn't dramatically having an effect. I mean, our results of the study itself showed that it's not having an effect, and if anything, we're seeing good results with this approach. And just to show you, a little bit more detail into, under 35 different age groups, you can see that the expected rate, if a patient stays in 3 cycles and they're under 35, it's an 84% live birth rate within 3 cycles. So, this is, we think, really good, promising data. It shows that the media that has a long shelf life is working well. So there's some issues. Let's talk about the implementation. Quality assurance remains critical, right? So you have to have, again, we talked about SOPs and these different pieces that I mentioned earlier. And I already had that slide. So this is a slide I wanted. Media management to practical realities. Now, this is where we're going to get into some touchy area. I'm going to really discuss the elephant in the room. So high volume clinics that have thousands of cycles use media rapidly, so it's not an issue. They just go through bottle after bottle. But low volume clinics, which is many of yours, you face waste. That's a challenge. And the problem is, after opening, you have this 1 week of having the media available. And if you try to keep it longer than that, you have risks because contamination, because pH rises as it equilibrates with the air, and you can have component precipitation. So this is the issue with, it's great to have a long shelf life, but you still, you need smaller bottles. So, the way that the manufacturer gets around making sure that things are good for that week is they're gas equilibrated. And then obviously they can't guarantee the pH. So this is the standard practice. Now, I wish I could take a poll, and didn't think of it before, a poll of how many people aliquot culture media out of the main bottle. And this is something that's been debated a lot in the US. I know a lot of groups that were doing aliquoting. I've never done aliquoting of culture media. But it is a common practice in lower volume clinics. At least I don't know if it still is. I'm putting it out there. It's the elephant in the room because I think it still probably goes on. If it's done properly, and it is a big if, because now you're going outside the certificate of analysis and the instructions for use by a few. But if it's done properly, you need to have test tubes that are validated for non-toxicity. I mean, that's really critical, add in toxicity when you put them in test tubes. You got to have good technique, single use only once you use that, proper labeling with expiry dates. And manufacturing shelf life does not automatically apply to aliquots. So that means that you've got to, we don't know once you've opened it and if you aliquot it, what's the shelf life then? So for something longer, you could say, well, maybe, instead of 1 week, maybe 1 month instead. And I'd be curious to hear what you guys say if you are aliquoting, how long you keep something before you consider it not usable. So the bottom line is, while it could be effective, it's generally not allowed. When I say not allowed, by scientific directors, but that varies. And I know there's economic realities to that. So, the best practice is you use manufacturer's bottles as intended, aliquoting only when operationally necessary. So I'll end on this future perspectives. We don't know anything about other culture media, whether they can go longer. It'd be great if manufacturers led some studies on that, because this could be a benefit. We've got our laboratory excellence that this is working well. I've demonstrated that through, the study we reported, as well as the one that I'm having reviewed currently. The industry impact is also good for sustainability goals through reduced waste and improved efficiency. And with that, I'll open it up for a discussion about how we optimize, our culture systems and laboratory management strategies. Great. Thank you.
Right. Thank you, Dean. That was absolutely wonderful. A very practical thing, let me put it honestly, but I read it twice to understand the paper. I had certain doubts. And I think this is not, I would not say groundbreaking, but a ground clearing paper. That one of the
Mm,
parameters has been completely cleared with the media. You know, it is if anything goes wrong, everybody is in a habit of blaming the media. So this
mm,
is something which has been taken care after reading and going through your paper. So kudos to that. Definitely, it's a wonderful job, and considering such a huge data is there.
mm.
But
Thank
with
you.
that, I would like to go in with certain questions that regarding the post handling, you mentioned about allocating, right?
Hmm.
So India, mostly, a country of saving, making cost effective.
Mm,
Many centers in India still believe in allocating. Also, the batch size in smaller cities, three-tier cities, is not big. So in such a scenario, allocating is quite a common practice. So obviously, it is done properly because the chances of contamination in smaller centers or any kind of centers is not that much as much information as I have. So in such a situation, how do you deal with this media? Like once you open the bottle, you keep it inside the incubator if you're not allocating, but do you put it back?
well, so we don't, so what would our practice, and this has always been my practice, is we'll have the stock bottle that'll be in the refrigerator. And we bring it out into the laminar flow hood, and we'll use something to aliquot it into
Yeah,
a vessel that we can use to then make dishes or do whatever. The bottle itself never goes in the incubator.
yeah,
And so, I think from what I've heard, and I remember this is what's crazy about all of this. There was actually, for a time, some of my peers in the US who said aliquoting was better. Like there's something, and so I was like, well, maybe I should. So I looked into it, and I didn't want to bother with it because it just felt like risk.
yeah,
Um, and I know that that's, there's realities that people have to deal with, and you can do it. But I'm not an authority on aliquoting, so I really can't speak to it. I can tell you what I would think and what I would do if I had to, and that would be, use a good laminar flow hood, use good technique, and just pray that you have tubes that are definitely not toxic.
right. So considering that this was only pertaining to one kind of media, different brands are there. So I love your slide regarding, the different companies. Some have 2 weeks, some have 3 months, some have 1 year. So if somebody is using a media which has the least amount of shelf life, and in such a scenario, the media, the manufacturer, from the date of manufacturing, it is coming to India, and then it comes to the center. So
Mm,
you lose
mm,
about 1 or 2 weeks. So you just
mm,
have a small
mhm.
shelf life. What your paper is designed is having a median of 6 months. If
Yeah.
it is at the fag end of the expiration date, and that is the manufacturer's validity expiration date. Is it okay and comfortable to use media even after the expiration date? If yes, why?
So, because I come from very regulated places, absolutely not. Like we would be in big trouble if we had any things that we were using that were expired. Um, so it might be best to leave it at that. If there was an emergency, it's
Yeah,
like,
obviously in
do you, do
emergency, I
you,
don't think nobody is
yeah.
going to.
Yeah, yeah. I mean, it's like milk, it's things you buy from the grocery store. They have their best buy date, but
Yeah,
you still use them after that. So it's possible, but in my worlds, it's not something that we can justify at all. So I really, yeah.
yeah,
I think the data speaks for itself, though. But the problem is those other ones, I mean, so my experience with GTL, which I don't know. GTL may be fine, and for some
yeah.
reason I only see myself, um, which is really bad. I have to change my view. Sorry, hold on. There. Uh, Suddenly I was staring just at myself talking. Um, is that I was using CSC. We like Life. We wanted to go to GTL. Did a test on it. I mean, that paper I showed there, which showed that, we didn't see an effect on our day 5 blasts. So we went forward, and then 3 months later, we found that our pregnancy rates weren't as good, and we switched back to CSC, and it was better. I think it was a pH thing at the time, but it's always something that you wonder about the ecosystem that we all use. Every one of our labs is a different ecosystem, and will something work as well in ours as someone else's? And so the same applies to, will the shelf life be the same? One thing that the both Cook media and the biomedics media have, and I think they're the only ones that are really high in osmolytes. So they have really high glycine and taurine, which makes them different than the others. So does that make the shelf life story different? Don't know, but we know that they definitely have that difference.
Okay.
Yeah, many congratulations, Dr. Morbeck. That was an excellent paper with
Thank you.
several strengths. I would just like you to comment on one aspect. In one of your limitations, you did mention that you did not account for post bottle opening handling. And probably if we look at one of your tables, looking at the G analysis, we can see that one site had a slightly lower odds in terms of live birth compared to the others. So can you, for the benefit of the audience, just extrapolate a little bit about what are the right practice post bottle opening, which will optimize results?
Right. So it's very good question. And it's a very technical one that I can speak to only so much, because with the number of labs we have and the fact that my role coming in here and only been here 2 years is not anymore at the bench. So I know what the SOP says, and the SOP, states what I said there, which is you work in the hood, exactly those steps. What we can't account for is whether there's any variation in that by other labs or within labs. And because we didn't track, we don't have any evidence to say, oh, we said for sure they all followed the protocols that way. I mean, it's not something, we do audits on people for things like ICSI and how they do a lot of different things. I don't know that we have an audit for that well enough to say that we can say for sure. Now, to the point of the one clinic that had lower rates, that clinic has been analyzed upside down and sideways and it cannot determine why it has a lower success rate. It's a very complex story. It's not got to do with the shelf life. It's not got to do with the media handling. It's something else. It's a strain that's a conundrum for me. I've spent many, many hours analyzing that one clinic. So, but it's not. It doesn't appear to be tied to this.
Okay.
So,
Yeah, please go ahead.
This particular, as you rightly mentioned, this particular paper was specifically on single step with time-lapse incubator. Would you think that you could compare this with conventional benchtop incubators, these results?
Good question as well. I've not given that thought, so I'm going to think out loud. It really should. I think it doesn't appear that the media would change. Now, let's just throw out there the idea that, well, if you're using say an old-fashioned big box incubator and you're not using reduced oxygen and you don't have a good gas line filter for your CO2, could there be an interaction that occurs if it's going off just a little bit, but it doesn't go off enough to show in the study? Hypothetically, that's possible. Again, it goes back to that ecosystem thing that it could be that in one lab that may have some effect. I think that's quite unlikely, but we can't rule it out. It's a very good point. Yeah.
All right.
And
And
just to add to that, in the same way, could these results be extrapolated to sequential media?
So that one, I think that they could, and because I think the difference is, so the sequential media, again, in the GEMS suite, and probably the Cook as well, because they're very similar. It's like these media have lineages, and CSC was a copy of Global, and this GEMS media was a copy of Cook. I mean, they came from each other. So I know that both of those media, both of the Cook sequential, have the high osmolytes as well. So I can say that they're probably going to do the same as the single step.
Okay.
Dr. Dean,
But the other sequentials in any other media, we can't say.
Dr.
Not
Dean,
not Vitrolife.
No way to know. Yeah.
so Dr. Dean, here I would like to ask, the media you have used, it has a very unique quality of 12-month shelf life, whereas many commercial media expire within 2 to 6 months. So do you
Mhm.
believe your finding extend to other brands as well? Or it is particularly formulation and manufacturing process of this particular media which is making it uniquely stable? So can we extrapolate these results with other brands as well? Compare
Yeah.
Indian
So,
steps.
I can't. So I'm always evidence-based. So to say it's possible with the others with the given differences, I can't say it is. I can put my theory out there, and I would guess that they probably are suitable for longer. And I don't know how though they make that change. Like I'm gonna guess that none of the media companies are all that interested in putting effort in to change something like this.
Okay.
Another question. So some of these bottles just contain one date, that is date of expiry. And if we look at your methods section, you do say that you spoke to the manufacturers to understand the date of manufacture. So since if you just received the media, how do we know how old it is? Do we have to call up the manufacturer or
I don't know for all other manufacturers. I mean, I had to do that in this case to know, well, how long is it before we get it? And, so we have that information in the manuscript, but I don't know if that's standard. We know Vitrolife is proud of and very good at doing a lot of QC. How much does that delay? Let me speak this time. It might be Sanjay.
I'm sorry.
Hey, Sanjay. Yeah, so it might be that theirs is longer before we get it because of the extra testing they do.
So do you think that, considering that now that instead of just the glutamine, you're having L-alanine and the dipeptide. So will other companies, do you think, are going to jump into this bandwagon because it is going to be more stable? Because what I saw is you are not replenishing the media. And the main reason has always been on day 3, we changed the media because of obviously ammonia is increasing. So is this the new deal, new solution that we are going to go in for undisturbed 5-day culture of embryos? If this has
So,
been the find, because this is a parallel finding I'm seeing, because
yeah,
this
yeah.
is a second conclusion I see out of this paper.
Yeah, you raise a good point. There has been a gradual change in a lot of the world to undisturbed, continuous, no media change. Now, when I was at Mayo Clinic, when we switched to global, we started with changing, and then we tested, is it better to change or not? And we found no difference. When I then moved to New Zealand, they were putting it in, giving the FERT results on day 1 and coming on day 5 and then doing a transfer or not, which to me, it's one reason I like time lapses, because you get that information without having to pull them out and look at them. So I know New Zealand's been doing that continuous since probably 2014. We started at Mayo in 2012. I think there's a lot of places in the world that are doing continuous without immediate change.
So is it safe to say that the dipeptide being used can make a difference, and we can use the media that has alanine glutamine combination for continuous culture? We don't need to change. Is it safe to say that? Would you agree with this? Would you recommend it?
So I think you have to test it in your system, which they also did here at Genea. So they similarly, when they went from sequential to continuous, they did changes and then they tested it. And I think every culture media now, I'm not aware of any that doesn't have the dipeptide. So they all should be suitable single step to culture throughout. But if I'm doing sequential, the first thing I would do is the side by side to say, how does this single step work? Once I'm in the single step with the change, I would then say, well, now how does it compare to do with and without change?
Dr. Dean, it was interesting that, all of us go on the premise that you want to use culture media, which is fresh and not later. But in your study, you had only 105 with less than 3 months. Because you didn't know you were planning the study at retrospective. So why was it that you had very few cases compared to the whole lot in the first 3 months?
Let's speak back to the other point of the shipping. We had to go to find out the manufacture date, and it's just how long it takes for them to test and then release and ship for us to start using it, and their production schedule. So it's in their production schedule.
Okay. I was also wondering the same.
Yeah.
And I have one more question. Dean, somewhere in your slides, you mentioned about media tracking systems. What were you referring to?
Well, there's many different ways to do it. So the different witnessing systems often have that built in where you can track lot numbers with that. The electronic medical records will also have systems where you could add that in, which would mean you'd have to write it on, you're writing it on every patient's chart and then putting it into the record, which is a pain and nobody wants to have to do that. We happen to have our own home system that's pretty simple, just similar to
So
a
you're
spreadsheet
just
system.
tracing the outcomes with the different lot numbers of media, right?
Correct. Yep. So we know when the media lot was changed, and these are the cycles that were done with that new lot.
Right.
Also, coming back to aliquoting, it's something which I've never been convinced about. Won't it also depend on the size of the container in which you're aliquoting? Because otherwise, there's going to be so much of a pH and osmolality change. So I mean, apart from, okay, we will say the pH will come back to normal in incubation, but there will be a change. So any kind of studies, I don't recollecting myself, but if anybody is aware of the dimension of the container in which you're putting. So if it's small Eppendorf tubes, maybe the changes may not be so much, but if you're going to use large, even 6 ml or 14 ml tubes, so that may bring about a lot of change in the pH osmolality by the time you use it. And, I mean, unlike you just get the large volume container out, take it, and then put it back immediately. I mean, if you're not incubating the medium. So any idea about that?
No,
because I don't think anybody's looked at it.
No, I don't think so either. And now another crazy thing that I just remembered is that one of the things some were doing in the US was rinsing the tubes before putting in because they were worried about contaminants in the tubes. So it's just, I never was fond.
That is interestingly weird.
So in that case, we'll come to a time where we'll have to get rid of plastics because there is so much of styrene in the VOCs.
Yeah.
When we are measuring it, and it's thanks to all our dishes and tubes and everything around.
Yeah.
So, I think a couple of people have questions, so maybe they can also ask it themselves or,
Dr. Somna, you wanted to ask something?
Yeah, just a small point. Since your studies went over about 3 years or so, you're likely to have used media in different batch numbers. So do you think that makes a difference? or are they all similar in terms of their?
Yeah, so that was a question from one of the reviewers. They wanted to know if there was any performance differences amongst the batches. And I thought, oh, that's a good idea. I'm going to go back and look. And when I looked, I saw that there was just no way the number of cases in each batch and then across the different units with different mixes, it became so heterogeneous in terms of how you would do the analysis. It wasn't possible to see.
There
No,
are a couple.
but
Yeah.
wasn't the whole idea behind media tracking is to make sure that there's batch to batch variability, right?
Well, I mean, yes. So for me, my number one reason is to say if suddenly I see a shift in outcomes, I want to be able to say, oh, the shift occurred that date. What did we change? We're tracking, we can say that's what changed. And then we can make it, investigate that further. That's how I look at it very practically. Regulatory wise, it goes back to the 90s when there was some issues with Irvine having some BCE. It was some HSA that might have been contaminated, so they had to do a recall. So there's more from things that might go into patients and need to be able to know what did that patient get exposed to. So there's a really regulatory piece that started some of it.
Dean, I really wanted to ask something about neonatal outcomes. Like when we used to use sequential media, there were probably 10 years back, there were quite a lot of paper which says that reduce birth weight with the older media uses in case of sequential media. But with your data in single step media show no such effect. So do you think these differences arises from media composition or from embryonic exposure duration or shift to blastocyst stage and the frozen transfers? How do you interpret these divergent neonatal findings?
Well, so that was the Kleiker's paper. That's the only one I'm aware of. If I missed one, I'll need to know. But I think there was just the one Kleiker's paper, which had only, a few hundred births. And so I think it's just underpowered to, birth weight studies are incredibly difficult. They need good sample sizes when you consider preterm births, multiples, and so on. So I think that was just probably an artifact. I don't think that was real.
So you are saying it may be because of other co-founding factors rather than.
Yes.
Okay. So there are quite few questions in the chat box as well. Dr. Gaurav has already asked that what kind of media tracking system.
Yeah, you can skip mine.
So Sonal really wanted to ask the time lapse incubator used by the clinics involved in this study were dry or humid. Does this study include this criteria as well? Humidity of
That's
the duct.
it's another ecosystem question that is very good. So does this apply if you're using dry culture? So this was all humid, humidified time-lapse incubation. So is media going to be as stable as long if it dries out? We don't know. Yeah.
So Dr. Sujata, she has complimented that it was a very nice presentation and she wanted some more information on post-opening of the media. Do you consider color change or 5-7 days?
The standard is pretty much like the conservative people will keep it 5 days, and I think the standard to me is more like 7 days. The media, the color will change, but if you're seeing precipitation, you shouldn't use it because the composition's different then. And it will start to precipitate if it gets really alkaline. And it's weird. I say it will precipitate. Not all of them do. There were some media that we had that would precipitate with when it went really basic and others that I wouldn't see precipitation. So
So there is another very interesting question by Dr. Satyavan. He is talking about different packaging. He is specifically talking about different companies are available in the market and they are using different capping. Some use plastic capping, other use rubber capping. So which one is the best for the media, rubber or plastic capping? What is your suggestion?
I don't know. All I know is I don't like the Cook rubber stopper capping. That to me was always a
Okay.
hassle. I don't know how that came about. They're the only ones that did. I think they're the only ones that do that.
Just to
In
conclude,
addition to just the same follow-up question, Rubber. So obviously nowadays we are having an option between glass bottles and plastic bottles. So some say that if it is glass, then there is zinc contamination. If it is plastic, then we have phthalates, we have bisphenols, we have everything. Which way to go? I mean, and also considering the environment, does it make sense? I mean, where are we?
Hmm. Great question, and I don't have that answer. I feel comfortable with both from a use standpoint. Like we're working and are we delivering good care? I think either one is fine. There's no evidence that there's a reason to be concerned. The environment and all the rest of the things, those are important questions as well, but beyond my pay grade.
So one last thing, Dean, the media you tested had a year's shelf life, while the others have been 60 to 120 days. And considering that you talked about 3 months time from manufacturing, testing, QC, and it reaches finally the clinic, does it become concern for media which have a shelf life of just 60 to 120 days? Because nearly quite a lot of time has gone by the time it reaches you. It reaches nearly the end of the shelf life for that matter.
Yeah. I can't remember which one it was, but I was using a media once upon a time where it seemed like it was pretty routine that we were having to replace it because the shelf life wasn't that long.
Yeah. But of course, your data shows, and it can be, I think we could extrapolate it to other media, then they possibly are in a safer zone, but then they have to extend their shelf life.
Yeah. I
Or
know.
print.
So this is the scary thing because we don't
Yeah.
know. So out of all of this, for me, and talking to all of you, knowing how driven you are to give the care and be able to keep costs down, yet what happens if you think, oh, this paper says that the shelf life can be ignored and it impacts outcomes, right? That's not
Yeah.
okay. And so we really need the companies to do their bit.
Yeah. Because normally the tendency is to look at the expiry date. And now you're making us look at the time of manufacture till the time it reaches you.
Yeah.
And that's quite long. I mean, I never really looked at it from that perspective.
Well, I don't think it matters. Their expiry
Yeah.
date is short, and therefore you look at the expiry dates. If they're only shipping like this one is, and there's only like a month or two of availability, you'll know that, and that really impacts your workflow.
Yeah.
So I think, Dr. Somana, you wanted to say something?
And last question, one last comment from you, Dr. Morbeck. Why is there so much secrecy about media composition?
No media company. I don't know. It's a good question. I've not heard a good reason, and I've talked to a lot of them, and there's nobody that's come up with like, well, no, it's going to be the most common one that I heard was like, well, no, then other companies will be able to copy them. Like, well, we already know we can do that. So I don't know. I think there might be something else. Maybe there's no idea.
Because if you extrapolate pharmaceutical drugs, I mean, everybody knows the composition. In fact, you cannot prescribe something whose composition you don't know. So it's a similar situation, and we have no idea about the composition.
I know.
At least some companies tell you the constituents. Some don't even tell us the constituents anymore.
I know. It's shocking. Yeah.
Yes. So I think we come to an end, and thank you. I know it's quite late there, and so nice of you to accept our invitation.
Well, thank you.
It's a very practical topic, right from, something we just take for granted, and a lot of thought-provoking. So when I saw that paper, it sort of opened your eyes to the discussion. And thank you, everybody, for joining in. And hope to see you in India sometime soon, Dean. Thank you so much.
Yes, I hope to be there sometime soon. I really enjoy my visits there. So
yeah, so thank you. And
great.
Thank you, everyone.
All right. Take care, everyone.
Yeah.
Thank you.
Good night.
Good
you.
night.
Thank
Thank you
you.
All
very
right.
much.
Good night.
Bye-bye.
Bye-bye.
Thanks,
Much.
everybody.
Thank you.
Thanks, everybody.